Okay, so here's what we're doing today. I'm gonna go through an introduction to DNA, just a PowerPoint lecture that I believe I already shared the lecture with you in the lecture section. So no notes to take. I'll just give you the filled in lecture, and then we'll just do a DNA booklet that will ask questions that are related to the lecture. So by the time we're done today, you should be able to describe the 3 roles of DNA, describe the smaller parts that make up DNA, and then we'll also talk about several different scientists that played a role in discovering DNA. So remember the organelle section that we already covered, that DNA is located in the nucleus. We'll talk about how it duplicates itself or replicates itself, and also how it passes on that genetic information to other parts of the cell. Okay, so here's the lecture right here. When we talk about the different roles of DNA, there's 3 things that DNA can do. And those 3 different roles are storing, copying, and also passing on the genetic information in the cell. All of that genetic information in the DNA starts out in the nucleus. And what you'll see the next couple of weeks, when the DNA duplicates itself, it's passed on to new cells that we call daughter cells. And when the DNA passes on the genetic information, it actually becomes RNA, and it transfers the RNA to different parts of the cell. And that's where those ribosomes come into play of putting proteins together. It's actually a pretty cool process when you think of how tiny the process is and the things that are happening with duplicating and replicating and passing on the genetic information to different parts of the cell. Okay, so the most important job is to store the information that takes place in the nucleus. The information is stored in genes, which control the pattern of development. Okay, so when we talk about that, we're talking about those instructions that cause a single cell to develop. They are written as a code into the DNA of each cell. And that code exists within the nucleus of the cell. What you'll learn today also is what parts of one DNA match up with other parts of another. And you might remember this from middle school, but that's when we start talking about the A's and the T's matching up and the C's and the G's. And you'll see what I mean by that when I show you a diagram in just a little bit. Cells divide. Remember on the Walk with Ryan movie, actually only one half of the cohort might have watched it, or one cohort might have watched it, but Ryan had to get out of the cell before the cell multiplied and divide. So cells divide, and when they do that, they make a complete copy of itself. All of the information in the cell is passed on to the new cell that forms, and we call that the daughter cell. Sometimes that information can get messed up a little bit. So we call those mutations in the DNA code. Okay, so scientists were studying this, and a lot of them didn't understand how the DNA was copied. So we'll talk about what happens with the copying process throughout this lecture also. Once the structure of the DNA was discovered, the mechanism for copying it was also discovered and understood. Okay, so here's your components of DNA, or the smaller parts that make up the DNA ladder. DNA is made up of nucleotides, and a nucleotide has 3 parts. Again, DNA is made up of a nucleotide, and the nucleotides are all chained together to form each side of the ladder, each side of the DNA ladder. Now, it's made up of 3 parts. All of the DNA, all the nucleotides will have a phosphate group, a 5-carbon sugar, and it's named that because it has 5 sides, 1, 2, 3, 4, 5, and a nitrogenous base. The nitrogenous bases are made up of the ATCGs. So if we ask you about the 3 parts of a nucleotide, a phosphate group, a 5-carbon sugar, and a nitrogenous base. All of these put together in this exact order, phosphate, sugar, base, phosphate, sugar, base, phosphate sugar base creates one side of the DNA ladder. When the bases are hooked together, that creates the other side of the DNA ladder. So let me show you that. So here are the nucleotides chained together. Phosphate group, 5-carbon sugar, nitrogen base. Phosphate group, 5-carbon sugar, nitrogen base. Phosphate group, 5-carbon sugar, nitrogen base. This would be one side of the DNA ladder. On the other side would be a nitrogen base hooked to it, a 5-carbon sugar, and a phosphate group. A nitrogen base hooked to it, a 5-carbon sugar, and a phosphate group. And when you put the other side together, that's the double helix. Now, these nitrogen bases have 4 different types to them. And I just refer to them as A, T, C, and G. The long name is adenine, cytosine, guanine, and thymine. And what you'll learn is that A's always pair up with T's over here, and C's always pair up with G's. And they're held together by a hydrogen bond. Okay, so here's the nitrogen bases and the bonds that hold them together. These nitrogen bases are always attached to the 5-carbon sugar, which is attached to the phosphate group. And you can see there are 4 different kinds of nitrogen bases, A, G, C, and T. So again, this would be just one side of the DNA ladder. The other side would match something with the A, which is always T, and would also match with the G, which is always C. These nucleotides can be joined in any order and any sequence of bases. That's what we call the genetic code. Your genetic code is made up of billions of these A, G, C's, and T's, and in a specific order to give you the characteristics that you have. Okay, so here's some of the scientists that led to the discovery of DNA. We refer to them as Chargaff, Franklin, Watson, Crick, and Wilkins. And you'll learn more about these people as we go through this unit right here. Okay, so the first one is old Erwin Chargaff, our good friend Erwin. Chargaff's rules says that the percentage of adenine A is always equal to the percent of thymine T. In other words, A pairs up with T. The same thing is said or can be said about G, guanine, and C, cytosine. So that's Chargaff's rules. If you have, listen to this, if you have 30% adenine, that also means you will have 30% thymine. So if you ever have to do an opener or a little worksheet or a quiz, and it says you have 30% adenine, how much thymine would you have? According to Chargaff's rules, the percent of adenine and thymine is the same. According to Chargaff's rules, the percent of guanine and cytosine is the same. Now, Watson and Crick just stated that, actually, Watson and Crick were wrong. They stated that A pairs with A's and C's with C's, etc. But we now know that A pairs with T and G pairs with C. Okay, so Rosie Franklin, good old Rosie, old Rosalind. She was a British scientist that used X-ray diffraction to actually get the structure of DNA. Now, there was a little bit of drama behind this picture. Rosie Franklin discovered this, but she was not a well-respected female scientist by the other male scientists. So Watson and Crick actually stole this photo. It's called the photo, I think it's called Photo 51. There's a movie that we usually watch when we're in class about this photo. So Rosalind Franklin discovered this double helix structure through the X-ray diffraction, but Watson and Crick stole it. Oh, here's the movie up here, The Secret of Photo 51. So if we come back face to face, we may watch that maybe the day before Christmas. But it has a whole background of how Franklin discovered it and Watson and Crick stole it and got credit for it. Okay, so this observation that A equals T and C equals G became known as the Chargaff's rules from Erwin Chargaff. This X-ray diffraction picture showed the X-shaped pattern that actually revealed that DNA is twisted around each other, like the coils of a spring. And that's where they got that double helix pattern from. Other clues from the picture suggest that those nitrogen bases are near the center of the DNA and they're hooked together by bonds. And on the outside of the DNA are the phosphate groups. Okay, so here's our good friends Watson and Crick. Around the same time that Rosie Franklin was using the X-ray diffraction to get the picture, James Watson and Francis Crick were also trying to understand the structure of DNA. The clues in Rosie Franklin's X-ray pattern enabled Watson and Crick to build a model, and that model explained the specific structure and properties of DNA. Now, the video that we watched, The Secret of Photo 51, goes into more detail, and it really sounds like Watson and Crick stole that photo. But really what's happening, I guess, with any type of science, and you can even say this the same for the vaccine for COVID now, is that anytime you have different groups of scientists trying to solve something, competition usually speeds things up because the scientists want to be the first one to discover it and get credit for it. And that's what Watson and Crick did, but they just used a little bit of Rosie Franklin's research to help do that.
All right, so Watson and Crick, when they were first starting out, they were model builders. So what they want to do is they would take things like this and they put them together and try to model out what it would look like. And so when they were trying to do that with the DNA molecule, they had Chargaff saying how the adenine were exactly the same, and the thymines were the same. So they came up with that idea that likes had to pair with likes. So they thought adenine and adenine went together, thymine and thymine went together, etc. So what that means is, like, if these are adenine, they go together. And so their original model they built is that they were kind of like together like this, and they stuck out. Okay, and so that was kind of how it would work. So you had adenine with adenine, and then like cytosine and cytosine, and guanine here. And so you had identical bases on either side. Well, then Rosalind Franklin came along, and she had her X-ray diffraction photo, that photo 51, or the B form of DNA that showed that X pattern. And I don't get that here, right? I don't get an X shape here. So like, okay, what about, wait a second, if they were like that, then they had to have connected on the inside. And so they're like, maybe we had our model wrong, and they flipped it around. Well, then when they did that, then if you can see like this, the problem was bases like adenine and guanine are actually longer than bases cytosine and thymine, which are shorter. So if you have an adenine and adenine on the opposite side, and say like a guanine down here, all of a sudden there's this huge gap. And that didn't work either because the two short ones weren't connecting. So then they had to come up with another rule. They're like, well, wait a second. If A and T's are exactly the same, maybe A doesn't pair with T at all. Or sorry, maybe A doesn't pair with A. Maybe A pairs with T. And then they started putting that together because T was a short base and A was a long base. And the same way goes, guanine is a long base and cytosine is a short base. So they started putting them together that way. And they're like, okay, maybe we got something here, right? And so then they started putting them together like that. So bear with me as I put these back together the way that they would have, right? So short base with a long base, etc. And so then you end up with this ladder. And then, of course, with the X shape, we know that it has to be twisted. So then when we twist, notice the X shape right there, right? So that is the X shape that we're seeing in that B form of the DNA, is where those cross over, because this ladder is then twisted, and that's what gives us our helix shape.
Okay, so from that little demonstration there from Coach Moore, just know that A's pairs with T and they have the same percent according to Chargaff's rules, and C pairs with G. And when they pair up, that forms two sides of the ladder, and the ladder is actually twisted into the helix.
All right, so Watson and Crick, when they were
Oh,
first...
darn it. Okay, so here's Watson and Crick's breakthrough model. Now look at this right here. Watson and Crick came up with the two strands wound around each other. But look at, this is what I want you to see here. The entire sides of the ladder are made up of sugar and phosphate. So we call it the sugar phosphate backbone. All of this is sugar and phosphate put together. And you can see it when we break down the nucleotide. Remember, a nucleotide is made of three parts: a phosphate group, a 5-carbon sugar, and an ATC or G, the nitrogen base. But also look at this. C is connected to G. A is connected to T. And that's the pattern you guys have to know. If I give you a 5-letter one side of the genetic code, you should be able to write the complement of that genetic code. It's called the complementary strand. So C goes with G, A with T, and G with C. Again, that's called the complementary strand. In this double helix model, the two strands, like we just showed you, twist around each other like spiral staircases. This double helix accounted for Rosie Franklin's X-ray and also explains Erwin Chargaff's rule of base pairing. So we're throwing out, listen, we're throwing out a lot of, or I'm throwing out, I don't know who we are, but I am throwing out a lot of names here. But really just know that Chargaff came up with the percent of bases equal to each other. Rosie Franklin had the X-ray picture, and Watson and Crick actually were the first to create the model that showed the double helix. Now, look at this right here. This is good. This is one side of the DNA ladder. Notice the nitrogen bases, G, A, T, and C. Those bases are held together by hydrogen bonds. So the other side of the ladder and the complement to the first side, C, T, A, and G. So again, this is what I was talking about. If I give you a code of letters, if I gave you C, 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 and I said, what's the complement to that? You should be able to tell me G, 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 G. Because C always pairs with G, and A always pairs with T. If I gave you AACC for one side of the DNA, the complement to AACC would be TTGG. So that's how the complementary base pairs work. And those are held together by hydrogen bonds. So we call the nitrogen bases the stair steps of the ladder, and then the phosphate groups and sugars would be the sides of the ladder. And this is actually twisted in a double helix. Okay, so there's the base pairing. That's what I've been talking about. A fits perfect with T, and G fits perfect with C. When we copy DNA and pass it on, these are the base pairs that to try to make an exact copy of the original DNA strand. And really, there's more to it. We'll get into it when we get into DNA replication. But what happens is this is actually unzipped down the middle. So one side, we unzip this down the middle, so we actually have two sides, which we can create two new strands from to be passed on to two new daughter cells. Okay, so the Watson and Crick realized that the base pairing explained Chargaff's rules, and it gave a reason as to why A matched with T and C matched with G. Okay, hold on a minute. I'm going to turn off my